Welcome to today's lecture in Introduction to Biopharmaceutics and Pharmacokinetics. In this lecture, besides learning the basic concepts, you will have more than 20 practice questions at the end. This lecture is based on the textbook Applied Biopharmaceutics and Pharmacokinetics, Chapter 1. You can find the contents from Access Pharmacy freely from our library. There are 7 learning objectives for this lecture. After the lecture, make sure that you are able to define the drug product performance and biopharmaceutics. Describe how biopharmaceutics affects drug product performance. Define pharmacokinetics and describe how pharmacokinetics is related to pharmacodynamics and drug toxicity. Define ADME and the term clinical pharmacokinetics. Define meanings of MEC, MTC, NDA, ANDA, API, TDM, Cmax, Cmin, Tmax, and AUC. Explain how clinical pharmacokinetics may be used to develop dosage regimens for drugs in patients. And finally, define pharmacokinetic models and list the assumptions that are used in developing a pharmacokinetic model. As we know, a new compound to be a drug needs to be developed and approved by the FDA. There's many steps involved in the drug development and approval process by the FDA. First of all, the new compound will be discovered and developed. Then, it will be investigated or studied in animals for the safety and possible or potential therapeutic effects. After this step proves that the new compound is safe and generates some potential therapeutic effects, then it can be approved to go to step 3, clinical research. In this step, the compound continues to be studied, but this time the subject to be studied is on human beings. After this step, bring the data that prove that the new compound can be safe on human beings and can generate therapeutic effects to treat certain disease. It can be approved by the FDA and can be released to the market. In the market, this new drug continue to be monitored to make sure the safety on the large population. For the step 3, it's very important to determine how the body does to the work, and pharmacokinetic is involved in all steps, but this step 3 is the most important thing because how the body of the human beings act to the drug is very important because that will determine the safety. In step 3, clinical research, there are 4 phases. Phase 1, the new compound will be tested in healthy volunteers at around 20 to 100 volunteers to determine the safety and appropriate dosage of the new compound for human beings. Then, if the phase 1 is successful and prove the safety and dosage of the new compound, then it will be continued to move to be tested on the patient, the real patients. For this phase of phase 2 here, the new compound will be tested on the efficacy and side effects to make sure that there's a balance or dominance of efficacy to compare with the side effects in the patients. If phase 2 is successful, then the new compound will be continued to go to phase 3. This time the drug is continued to be tested on patients, but the number of patients increasing. It can be multi-centers in different institutions, different places. This time, the compound still be tested about efficacy and side effects, and also monitor the adverse reaction to some certain, kinds of patient with different critical condition. If the phase 3 is successful, the drug can be released to the market, and usually. This phase 3 here, after phase 3, the drug can be released to the market and it still continue need to be tested on patient, larger population. And this time is we call phase 4 or post-marketing. And this one, several thousand volunteers with different disease. For this phase, they can find the side effects or rare side effects happen to certain subjects. And in the 4 phases here, for the clinical research, the pharmacokinetics only required for every step. So a new compound after being approved by the FDA can be a new drug. And the drugs as substance is used not only for the treatment, but it can be some drugs used for the diagnosis, mitigation, or prevention of disease. Drug usually is put in a dosage forms or delivery system, and the whole system there we call drug products. The drug products can be in solids, semi-solids, or liquids. For the solids, we have tablets, capsules. For the semi-solids, we have ointments, creams, gel, etc. For the liquids, we have solution, suspension, and emulsions. Drug products can produce a systemic effect for the whole body or just local therapeutic effects such as topical, some topical drugs, some pain release at the topical site. The important qualities of a drug product is the safety, then effectiveness, and convenience. So these are the 3 important things that determine the qualities of the drug. Then another terminology you must know is a drug product performance. Drug product performance is the release of the drug substance from the drug product, and that leads to the bioavailability of the drug substance, and then eventually lead to the pharmacologic effects, and these effects can be desirable or undesirable. The pharmacologic effects that is desirable, usually we call therapeutic effects. If it's an undesirable effect, we call side effects, or sometimes it can be the toxicity. What is biopharmaceutics? Biopharmaceutics is the study to evaluate the interrelationship of the physical chemical properties of the drug, of the dosage form in which the drug is given, and the route of administration, and the rate and extent of systemic drug absorption. So you learned already about pharmaceutics. Pharmaceutics is the study of the physical chemical properties of the drug and the drug dosage forms. When the drug is given to the body, the biopharmaceutic will cover not only pharmaceutic but also what happened, what's the relationship between the drug itself, the drug product itself, and the route of administration, and then how the drug can appear in the blood or in the systemic circulation after the absorption. So biopharmaceutics study deeply not only in the drugs outside the body, but also when the drug go inside into the body. Describe the relationship between drug product, and pharmacologic effect. Drug is the new compound approved by the FDA. Drug product is the system contain the drug, or usually we call drug dosage forms or delivery system. And pharmacologic effects is the effects caused by the drug from the drug product. So we know that when the drug under a dosage forms is taken by the patient, it will be administered through, it can be from oral administration, it can be from injection administration, etc. For example, for the oral, if it's a solid dosage forms, the drug will be disintegrated and dissolved. And from there, the drug will be released out of the dosage form and will be absorbed into the body and reach the systemic circulation. From the systemic circulation, the drug will move to the tissue. And from the tissue, the drug can reach the site of action, react with some receptors, or from different mechanism of action, the drug will generate a pharmacologic effect or clinical effect. Between the systemic between the systemic side and the tissue side, there's always a movement of the drug between two sides until it reaches the equilibrium state. At the equilibrium state, the drug will be excreted, or it can be metabolized after metabolism, it can be excreted. And this combined process of excretion and metabolism is called elimination. For the when the drug reaches the tissue here, the drug can cause some pharmacologic effect, but to be considered clinical effects and have some effect real to treat the disease or have some preventive action, the drug needs to reach certain concentration. We call the minimum effective concentration. And usually you cannot measure the drug at the site in the tissue here. And only site the drug can be measured, the drug concentration can be measured is the systemic circulation, that's mean in the blood. And from here, the concentration of the drug will be measured, and then from certain concentration, it will be associated with the pharmacologic effect or clinical effect. And the measurement of drug in the blood here to predict the action of the drug in this size here is a case of pharmacokinetics. And the time when the drug release and in the dissolution before it can be absorbed in here, this phase we call biopharmaceutics. And the phase that the drug in the tissue generate the clinical effects we call pharmacodynamics. In pharmacokinetics, for process very important is absorption, distribution, metabolism, and excretion. Any drug is administered to the body will be absorbed to the bloodstream system here. And from here, there's some kinds of binding with protein from the drug can happen with certain drug. And then the drug will be distributed to the tissue as I mentioned before. And then when it's reached the equilibrium state, the drug will be excreted or eliminated out of the body. And also before the drug reach the bloodstream here, the drug can have some kinds of metabolism through the liver, and it can create the metabolites and can be eliminated out of the body. And the process of elimination is including metabolism and excretion. Another process we call disposition is the combined process of distribution and elimination here. We call the disposition. So how biopharmaceutics can affect the drug product performance? As we know that biopharmaceutics is to study the interrelationship between the physical, chemical properties of the drug in the drug dosage forms and the routes of administration to the rate and extent of the drug when it's absorbed into the systemic circulation. So the dosage form style like solid, semi-solid, or liquid can affect the drug product performance. Physical chemical properties, for example, hydrophilic, lipophilic, amphiphilic, or some other solubility its own can influence the drug product performance. Route of administration can also influence the drug product performance because different route of administration like oral, injection, or directly injection, IV intravenous injection, subcutaneous injection, etc. It can influence to the drug product performance. And when the drug product performance is influenced, it can influence to the rate and extent of the systemic drug absorption. How the pharmacokinetics is related to pharmacodynamics and drug toxicity. Pharmacokinetics is to study how the body acts to the drug, how the body does to the drug. Or pharmacokinetics study the drug absorption, distribution, metabolism, and excretion. Pharmacodynamics is to study or describe how or what the drug does to the body. This means how the drug can generate the pharmacologic effect or clinical effect to the body. Pharmacokinetic, we call PK, or pharmacodynamics, we call PD, has a strong relationship. So how it has some relationship? In pharmacokinetics, any site of drug administration can influence the drug absorption, distribution, metabolism, and excretion. The drug property can also influence to its distribution in the tissue. For example, digoxin is highly affinity to the cardiac tissue. When from the systemic circulation, the drug can be distributed in the tissue and stay long there. However, aminoglycosides antibiotic drugs is a kind of very hydrophilic drug and doesn't have much like kinds of affinity to the tissue. The distribution of the drug in the tissue is totally different. So the pharmacokinetics property and properties of drug has highly influence of the presence of the drug in the tissue. From pharmacokinetics, if the drug is not absorbed well, just small amount of drug, a small concentration of drug appear in the systemic circulation will result in the consequence that not a lot of drug in the tissue. And when small amount of drug in the tissue, even though it can generate some pharmacology effect, but the effect is very minimal. It's not like considerable, or it cannot be strong enough to have clinical effect to treat the disease or to prevent certain kinds of disease. So pharmacokinetic, the drug in the pharmacokinetics, for what reason it needs to stay? If it can stay in the circulation, it can distribute to the tissue to generate the therapeutic effects. It's very important. If the drug has very good absorption, it can appear high concentration here. And with the good concentration, enough concentration, when it generates the therapeutic effects by bind to receptor, react to receptor, it can generate a high intensity. And it can have enough time, enough strength to cause the clinical effect. In case, if the drug, for some reason, it just like suddenly appear, it's large amount, high concentration in the circulation, for example, for like overdose or for some kinds of drug-drug interaction, it has some abnormal high concentration in here. More drug will be distributed in the tissue, and when it's with a high concentration in here, it can have very high intensity and tight course of drug effect. And with the strong effect like that, it may bypass the limit of the body to handle, and it can cause toxicity. The best way to know when the drug concentration in tissue reach certain intensity to cause the right therapeutic effects is to measure the drug concentration in the tissue where the site of action happen. However, it's not possible because you cannot measure the drug concentration in the tissue of the patient. There's only way is to measure the drug concentration in the blood or in the systemic circulation. And from there, from the concentration of drug there, we can predict the concentration of drug in the tissue. So a drug, for example, if it's taken orally, the drug needs to go through the GI tract, liver, and reach systemic circulation, right? So when the drug is given to the patient at a certain time point, the blood from the patient, the blood will be withdrawn, and then the drug concentration from the blood will be quantified. And usually they use plasma or serum to measure the drug. They don't use the whole blood because whole blood is very difficult. It's very difficult to quantify any drug in the whole blood because there's so many ingredients in there, components in there. It can interact with the assay. So after they draw the blood from the patient, the drug usually will be like centrifuged to obtain the plasma or serum, and then from the plasma or serum, normally plasma will be measured with the drug inside. And every time point, usually, for example, at 1 hour, 4 hour, 12 hour, 8 hour, etc. It depends on different kinds of drug. The patient will be withdraw the drug, and then withdraw the blood, and then the blood will be quantified for the drug inside. So any time point, the drug will be quantified, and then the concentration of drug and the time will be plotted in a graph we call plasma concentration versus time curve. And the time curve has the X bar to represent the time and the Y bar to represent the drug concentration in the plasma. This graph here is to describe, just like typical example, the plasma concentration versus time curve, or the PK curve, PK profile of an oral drug. The drug, when it's given orally, it has to go through different process before it's reached the systemic circulation. So here it takes time for the maximum concentration of drug will be in the blood system, and this here maximum here. And when there's a equilibrium state between the circulation system and the tissue, then the drug will be excreted or eliminated. Then the drug concentration in the blood will be decreased. And the decrease here will happen like gradually. The maximum of the drug to reach the concentration here, the maximum of the drug in the blood we call Cmax. And the minimum of drug can appear in the blood we call Cmin. Any drug to generate a therapeutic effects, they will measure, and when the therapeutic effects occur, they will quantify they will estimate the concentration to have the pharmacologic effects or clinical effects. And the time that the drug reach the minimum effective concentration or minimum concentration that can cause the clinical effects, the time here we call the onset time. And the first concentration of the drug, that the minimum concentration of drug that can cause therapeutic effects, we call MEC, or minimum effective concentration. The duration between the onset time and when the drug, when it's go down, reach the MEC here, we call the duration of effect. And the distance here between the MEC and the maximum concentration here we call intensity. Certain drug, when it's overdose or for some reason the dose is high, or for some condition the drug concentration in the blood suddenly higher than expected, it can reach to certain higher concentration than expected. And when it's too high, the high concentration here, it causes more drug in the tissue, more therapeutic effects, and it can over certain limit and can cause toxicity to body. The concentration that the drug can start causing the toxicity to the body we call minimum toxicity concentration, or MTC. And the distance between the MEC to MTC we call the therapeutic window. If a drug has a very difference between MEC and MTC, we call the drug has a large therapeutic window. If the drug has very narrow, not much different between MEC and MTC, we call the drug has a narrow therapeutic window. And with the drug we having narrow therapeutic window is very dangerous, because anything like just sudden increase the concentration a little bit, it can bypass the MEC and can reach the MTC and can cause toxicity to the patient. And for most of the drug here, with the narrow therapeutic window, it's necessary to be monitored on the patient and monitor the concentration, how the drug absorbs, how the blood, and they need to monitor the blood in the body of the patient to make sure that when they measure the drug, to make sure that they don't want the concentration of drug bypass the window here. And the monitoring of this, apply the pharmacokinetics concept, apply the pharmacokinetics method to monitor the drug to make sure that the drugs range within MEC and MTC is a case of we call clinical pharmacokinetics. And to estimate or to calculate the drug, the whole drug appear in the body, absorb to the body, we measure the whole area under here, under the plasma concentration versus time curve here, and that we call AUC, or area under the curve. The area under the curve is helpful to determine the extent of drug is absorbed in the body or the bioavailability of the drug. So as I mentioned before, pharmacokinetics relate to the drug substance when it's absorbed to the systemic circulation. So this is pharmacokinetics. And we usually, how to determine the pharmacokinetics of the drug, we build or establish the plasma concentration versus as time curve as in the previous slide. For pharmacodynamics, that means this stage the drug reach the site of action in the tissue and reach the site of action and cause some pharmacologic effect already. For the pharmacodynamics, how to monitor the drug, they build the kinds of a curve. It's different curve. It's a curve to represent the efficacy of the drug with the dose or the concentration of the drug. And when pharmacokinetics and pharmacodynamics combine, the curve will be called PK-PD. This is a relationship between the dose and the effect of the drug versus time on the patient. So ultimately, PK and PD is combined together to determine what dose can have the effect on the patient for how long with the time. So this is the ultimate goal, to have PK-PD parameter or PK-PD information on the treatment, on the regimen of treatment for the patient. So from the plasma drug concentration versus time curve, we can predict the drug concentration on the tissue, and from there we can predict the therapeutic effects. However, different drug has different property, and even different dosage form can create different properties of the drug regarding the distribution, absorption. So to facilitate the prediction from the drug concentration versus time curve to the therapeutic effects or pharmacodynamic effects, and sometimes it can possible toxic effects, they create pharmacokinetics models. The model is not a real one, it's just hypothesis using the math terms, math tools to describe the quantitative relationship between the time course of the drug in the body versus its PK or toxic effects. And this model also is very effective in describing the basic picture of the product design or the dosage regimen on the action of the body on the drug. Generally, there are three models, three basic PK models. However, two models as recognized or used or mentioned the most is physiologic and compartment models, in which the compartment models is widely used or commonly used in the clinical pharmacokinetics. Now we go to detail about PK models. The first one is a physiologic model or physiologically based PK modeling or PPPK. For this model is based on the actual tissues and their respective blood flow. The organs or tissue with similar blood perfusion rate and lipid content will be grouped together. For example, there's a group of heart, muscle, slowly equilibrating a tissue and rapidly equilibrating tissue, kidney, liver. So with different groups like that, and each box will represent a tissue compartment. So from this one, for each tissue compartment here, the size and the mass is determined like physically based on the real anatomy of the body rather than by the mass estimation. So this model can be applied to different species like mouse, rats, monkeys, or even like some kinds of dogs. So for this kinds of species, they will measure the size and mass. And the drug concentration in the tissue, of course, they still need to measure the drug concentration in the blood. And from there, they create this model. And drug concentration of tissue is the health kinds of determined by the ability of the tissue to accumulate the drug, as well as by the rate of blood perfusion to the tissue. And the blood perfusion to the tissue is represented by the letter Q. So we have, like in the model here, you see there's a lot of Q, different Q, because the blood perfusion to different tissue can have different rate. And then for certain organ like kidney or liver, when the drug is eliminated, they also measure the rate constant of the drug eliminated, the called K. So in this case, a very complicated model is generated still using the math. For different Q and K, they generate from using a software to make sure that like there's a relationship between drug in the blood and the Q and the K to different model like that. And they generate a model or template of formulation to predict the concentration of from the concentration of the drug in the plasma to predict the therapeutic effects of the drug in the tissue. And these models can be applied to several species, and sometimes it can be extrapolated from the animal data to the human data. However, it's very complex and difficult to use for general application. This one's more in like research or rather than like use in practice for clinical pharmacokinetics. The most commonly used model in calculate the drug concentration and predict the concentration is the compartment model. This compartment model is not a real physiologic or anatomic region. It's totally like mass, using mass. It's considered like tissue or group of tissue that have similar blood flow and drug affinity. It's very simple and easy to use for general application, and for this model, it cannot be used to extrapolate from animal data to human beings. Everything is constructed in the compartment model is using the mass tool. And in the compartment model, there's two very typical model. Is a one compartment model and two compartment model. For one compartment model, for the drug follow one compartment model is considered like well perfusion, like well distribution, where it's injected to body or distribute or administered to the body, almost like immediately the drug will be distributed to the whole body. That's the assumption of this drug, and usually this happens with some drug has very good like perfusion rate of blood flow and it can be distributed to the whole body. For the two compartment model, it's a little bit more complicated to certain drug. Like certain drug, when it's injected or administered to the body, what happens? It will distribute it to certain organ first. And because of the properties of the drug, maybe it has high affinity to the tissue, or it has some kinds of high distribution to the tissue because of the drug property. For example, the drug has highly distributed to the brain, to liver, or to lung. So in this case, for this kind of drug, it will be like first, it will be mostly distributed to certain organ first, and then it takes time before it can be distributed to the whole body. So for this two compartment model, it seems like two phases. The phase one, it needs to reach the affinity organ first. And the phase two, with time, it will distribute it to the whole body. The one compartment model. Now we go to the detail of this compartment model. This compartment model represents plasma and highly perfused tissue, and mostly like the drug is like distributed very quickly, highly perfused like to liver or kidney. And when after it's reached a certain equilibrium state, drugs will be eliminated from the central compartment. The two-compartment model. Usually, the central compartment model for two-compartment model is the plasma. And then tissue compartment, like on the tissue. The drug can be moved between central and tissue compartment, and the drug eliminated is still from the central compartment. This is some example about the model they use to represent the compartment model. Just like very simple, they use the box to design. Like for model 1, it's one compartment model with the IV injection. And this is model 2, one compartment open model with a first order absorption. That means normally it's oral absorption, extravascular administration like oral. It can be oral, it can be transdermal, it can be some kinds of subcutaneous injection or some kinds of intramuscular injection, but it's not the injection like directly to the blood. They call extravascular administration. Model 3 is about two-compartment open model for IV injection, and Model 4 for two-compartment open model with first-order absorption or oral or extravascular administration. For the model here, in general compartment model, they use only one parameter to measure is the K. For the model 1, K, this means the elimination rate constant, or the K0 is the absorption rate constant. Now let me give some more detail about the model for one compartment model first. Now we go to model 1. This model 1, when the drugs is like distributed quickly, perfused quickly, distributed quickly in the body, this drug is very simple. It's like only one compartment like this, and then this is kind IV injection, so there's no like a step of oral administration or like onset of action or something. The drug go directly to the blood, and this is the blood or circulation system they call central compartment, and the drug is introduced directly to central compartment, and then the drug later will be eliminated from the central compartment. And the parameter to describe the process of drug elimination out of the central compartment is a K. K is like elimination rate constant. The model 2, a little bit more complicated. When the drug is not directly go to the central compartment, it need to go through the process of oral absorption before it reach the central compartment. But for this drug, the drug also be eliminated out of the body from the central compartment. And for this model, two parameters are established to represent the process. The first one is K0. K0 is the absorption rate constant to measure how fast the drug can be absorbed to the central compartment. The second parameter is a K, or sometimes they call KE or KEL. That means the elimination rate constant to describe how fast the drug will be eliminated out of the body. For the two-compartment model, it's a little bit more complicated. Model 3 here. This is a two compartment. This is a central compartment, and this is a tissue compartment. One is central and two tissue, and they're always the moving between the central and the tissue in the model, and the drugs always is eliminated through the central compartment. And for this model, they measure the K is the elimination rate constant to measure how fast the drug is eliminated from the central compartment. However, they also measure the rate constant of the drug movement between the two compartments, 1 and 2, with K12 is the rate to measure the drug from compartment 1 to compartment 2, and K21 to measure the rate of the drug from compartment 2 to 1. Model 4. Model 4 is still a two-compartment model, but the drug is administered through extravascular and follow the first order absorption. And in this model, similar to model 3, they have to measure K12, K21. For this model, because there's a drug absorption, they have to measure the rate constant of drug absorption too. So the compartment model generally everything is like hypothesized and created using the mass, and it's very simple. And there's a software to help, like for whenever the drug, the blood or plasma is collected, the drug is quantified with the concentration inside. They build the plasma concentration drug versus time curve. Then they have the model with kinds of formula to calculate, and there's software to help to facilitate the calculation. And from this one, the K or K12 or K0, etc., will be calculated easily. And from the K value, the drug of later parameter pharmacokinetics will be calculated, and finally, there will be equation to calculate the drug concentration, the therapeutic effects, and etc. How clinical pharmacokinetics may be used to develop dosage regimens for drugs in patients. So from like when this new drugs is approved by the FDA, so on the drug we have all the information about drug safety, drug effects, and different information. And then the drug approval they need to be labeled based on the FDA requirement. And the labels generally will be inserted to the package delivered to the patient. And the package insert is used by the professional, by the doctor, pharmacist, professional healthcare professionals, and also is provided to the patient. We call the package insert. And this one, the information of the drug in the package insert is applied for general population. So sometimes some patient may respond differently. So that's why certain drug, even though it works for most of the population, but sometimes it can be toxic. It can cause some like undesirable effect for certain patient with different condition. So the application or the use of pharmacokinetic approach to clinics to individualize, optimize the drug dosage form, to optimize the drug dosage regimen, and to optimize the therapeutic effects on the patient, we call clinical pharmacokinetics or clinical PK. So for the clinical PK, there's a lot of parameter, there's a lot of formula used, and usually clinical PK, when before they work in the patient, they need to base on the population PK. Population PK is they study the PK of the drug in different population of patients. As you know that in clinical trials, they have phase 1, phase 2, phase 3, right? So from that population, the PK is generated from average population they study, and that parameter is used to apply to like individual when they treat the patient in clinics. And usually clinical PK is required for on the clinical research of a new drug. And after the drug is on the market already, you know that for some drug with narrow therapeutic windows, it's very dangerous because any change in the drug concentration in the plasma, it can lead to the change in the therapeutic effects, and it can cause toxicity. In this case, like clinical PK is very important, and it is required to monitor the drug concentration or the therapeutic drug to the patient to make sure that the drug concentration always in the therapeutic window or always between MEC and MTC. And it happens with most of the drug, like for antibiotics, anticonvulsion, some kind with a transplantation, etc. How prescribing information or approved labeling for a drug helps the practitioner to recommend an appropriate dosage regimen for a patient. Now look at the example of package insert of oral vancomycin here. You need to check out this package insert and look at the data of oral vancomycin from the drug dosage form, administration to the pharmacokinetics information. Then we will discuss in class. In summary, after you finish this lecture, make sure that there's some kinds of very, very basic information you need to know about the terminology of drug product performance. This is the release of the drug substance from the drug product, leading to bioavailability of the drug substance and eventually lead to the pharmacologic effects or clinical effects. Biopharmaceutics is study or evaluates the interrelationship of the physical, chemical properties of the drug product, and the route of administration on the bioavailability of systemic drug absorption. Pharmacokinetics is the study of how the drug or what the drug can act or can do to the body, or study the ADME, absorption, distribution, metabolism, and excretion. Pharmacodynamics is the study of how the drug acts to the body or what the drug can do to the body. Clinical pharmacokinetics is the application of pharmacokinetic methods to drug therapy in patient care or apply the PK methods to optimize the dosage regimen to treat the patient in the healthcare. Clinical PK is very important in therapeutic drug monitoring for potent drugs or the drug has very narrow therapeutic window. Two major pharmacokinetic models are physiologic model and compartment model. Both models are hypotheses and need different assumption and mass modeling to establish. The model are used hypothetically relate to the time course of the drug in the body to its pharmacodynamic and or toxic effects. These are basic abbreviation of terminology using biopharmaceutics and pharmacokinetics. You must know of MEC, minimum effective concentration, MTC, minimum toxic concentration, NDA and ANDA, API, TDM, Cmax, Cmin, Tmax, and AUC. After this lecture, you have more than 20 questions to practice. This is to strengthen your knowledge as well as your understanding about the lecture. Question: which phase of FDA drug development approval process held healthy volunteers? A. Phase 1, B. Phase 2, C. Phase 3, D. Phase 4. This question makes you to recall the understanding of the clinical phases, clinical trials according to the FDA drug development approval process. The clinical trials start with the phase 1 in which healthy volunteers are involved. This phase is to identify the dosage and also to identify or investigate the safety of the drug on healthy volunteers. After the phase is approved, the drug can be moved to the next phases, and next phases are involved with patients. Question: which of the following terminologies refers to the release of the drug substance from the drug product, leading to bioavailability of the drug substance and eventually leading to pharmacological effects? A. Drug products, B. Drug product performance, C. Dosage forms, D. Drug delivery system. This question makes you to recall about the definition of terminology for different terminologies such as drug products, drug product performance. So make sure that you come back to the slide to check your correct answer. Question: which of the following terminologies refers to evaluating the interrelationship of the physical chemical properties of the drug dosage forms, the route of administration, and the rate and extent of systemic drug absorption? A. Biopharmaceutics, B. Pharmacokinetics, C. Pharmacodynamics, D. Pharmaceutics, E. Clinical pharmacokinetics. This question makes you to review the definitions of basic terminologies using the pharmacokinetic studies. Make sure that you distinguish the difference between pharmacokinetics and pharmacodynamics, between biopharmaceutics and pharmaceutics, or between pharmacokinetics and clinical pharmacokinetics. Make sure you check the slides for the correct answer. Question: Which phase of FDA drug development approval process is mainly for investigating the efficacy and monitoring of adverse reactions? A. Phase 1, B. Phase 2, C. Phase 3, D. Phase 4. This question makes you to review the understanding of the purpose of each clinical trial phase from phase 1 to phase 4. Phase 1 is on healthy volunteer, mostly for investigating the safety as well as the dosage of the drug. Phase 2 is on the patients, with like on the patient in the small samples of patients, and this one mostly used to investigate efficacy and as well as some possible adverse reaction. And phase 3 is continue to investigate efficacy and monitoring of adverse reaction. For this CAP question, you should be able to answer within 10 to 15 seconds. After this question, from now on, I will give you the question and with the timer for you to practice. Question: which of the following terminologies refers to the application of pharmacokinetic methods to drug therapy in patient care? A. Biopharmaceutics, B. Pharmacokinetics, C. Pharmacodynamics, D. Pharmaceutics, E. Clinical pharmacokinetics. You have 15 seconds to answer this question. Question: this is a fill in the blank question. You have empty blank. Then describe what the body does to the drug. A. Biopharmaceutics, B. Pharmacokinetics, C. Pharmacodynamics, D. Pharmaceutics. You have 10 seconds to answer this question. Question: you need to select the following choice to complete the sentence. You have empty in front. Then describe what the drug does to the body. A. Biopharmaceutics, B. Pharmacokinetics, C. Pharmacodynamics, D. Pharmaceutics. You have 10 seconds to answer this question. Question: at what concentration that a drug can start producing a pharmacodynamic response or therapeutic effects? A. MEC, B. MTC, C. Cmax, D. Cmin. You have 10 seconds to answer this question. Question: which of the following processes are included in ADME or ADME? Select all that apply. A. Absorption, B. Distribution, C. Metabolism, D. Excretion, E. Reaction. You have 15 seconds to answer this question. Question: which factors can importantly affect the drug product performance? Select all that apply. A. Routes of administration, B. Physical chemical properties of the drug or drug product, C. Dosage forms, D. Colors of the drug or drug product. You have 20 seconds to answer this question. Question. Which of the following graphs closely describe drug pharmacokinetic profiles? Select all that apply. This is a multi-selection question. You will have more than one correct choice. So from the graphs, you have 4 graphs, A, B, C, and D. For this question, you have 15 seconds to answer. Question. Which of the following models are basic pharmacokinetic models? Select all that apply. A. Physiologic model. B. Catenary model. C. Compartment model. D. Non-compartment model. You have 20 seconds to answer this question. Question. Which of the following properties or description may not relate to pharmacokinetic models? A. Hypothetical model. B. Mathematical model. C. Relating the time course of drug in the body to its pharmacodynamic effects. D. Real models in human body. For this question, you have 30 seconds to answer. Question. Which of the following graphs represent a compartment model and a PBPK model respectively? You have 2 graphs, A and B, so name A and B respectively, either for compartment model or for PBPK model. PBPK is a pharmacologically based pharmacokinetic. You have 10 seconds to answer this question. Question. Which of the following properties or descriptions closely relate to the PBPK modeling? Select all that apply. A. Most frequently used in pharmacokinetic study. B. Based on actual tissues and their respective blood flow. C. Can be extrapolated to human data for some drugs. D. Simple and easy for general application. E. Group organs or tissues according to RET and SET. You have 30 seconds to answer this question. To answer this question, you need to come back to the slides that have description about PK model, and also make sure that you know the difference between the PBPK modeling and the compartment modeling. Question. Which of the following properties or descriptions closely relate to the compartment modeling? Select all that apply. A. Most frequently used in pharmacokinetic study. B. Based on actual tissue and their respective blood flow. C. Can be used to extrapolate from animal data to human beings for some drugs. D. Simple and easy for general application. E. Group organs tissue according to the RET and SET. RET means rapid equilibrating tissue, and SET is slowly equilibrating tissues. This question is similar to the previous one, so you have 15 seconds to answer the question. Question: which of the following properties or descriptions closely relates to the image below? A. One compartment model. B. Two compartment model. C. Three compartment model. D. Four compartment model. E. PBPK model. You have 15 seconds to answer this question. Question: which of the following properties or descriptions closely relate to the image below? Select all that apply. A. The central compartment represents plasma and highly perfused tissues. B. The central compartment represents plasma. C. The peripheral compartments represent tissue. D. Drug is eliminated from the peripheral compartments. E. Drug is eliminated from the central compartments. F. Drug is absorbed from oral administration. G. Drug is administered by intravenous injection. H. Drugs can move between central and tissue compartments. You have 30 seconds to answer this question. To answer correctly on this question, make sure you come back to review the compartments description for different kinds of compartments and properties. Question: which of the following properties or descriptions closely relate to the image below? Select all that apply. A. The central compartment represents plasma and highly perfused tissues. B. The central compartment represents plasma. C. The peripheral compartments represent tissue. D. Drug is eliminated from the peripheral compartments. E. Drug is eliminated from the central compartments. F. Drug is absorbed from oral administration. G. Drug is administered by intravenous injection. H. Drugs can move between central and tissue compartments. You have 30 seconds to answer this question. Question: what are clinical pharmacokinetics mainly for? Select all that apply. A. To customize and optimize dosing strategies for specific patients based on patients' specific diseases and conditions. B. To study the pharmacokinetics of drugs on different population of patients. C. To be applied to therapeutic drug monitoring for potent drugs. D. To customize and optimize dosing strategies for animals based on their specific diseases and conditions. You have 30 seconds to answer this question. This question relates to the concepts or definition of clinical pharmacokinetics as well as the usage of clinical pharmacokinetics. Question: name the boxes 1 to 8 shown in the graphs below. You have 30 seconds to answer this question. This question makes you to review your understanding of the pharmacokinetics profiles based on the plasma level versus time curve. So for these graphs, for those two graphs, make sure that you understand the meaning, the point of type of onset of action, Cmax, Tmax, intensity, duration, MEC and MTC and AUC. Question: matching the abbreviations and terminologies from the table below. You have abbreviations in one column and terminologies in another column. Make sure that you match appropriate terminologies for each abbreviations of MEC, MTC, NDA, ANDA, TDM, Cmax, Cmin, Tmax, half-life, AUC, and API. You have 30 seconds to answer the question. That's all for this lecture. I hope that you have learned some basic concepts of introduction about biopharmaceutics and pharmacokinetics. Thank you for your attention.